morning, everybody. I work in the pharmacology lab at the University of São Paulo. We are focused in the detection on new genetic factors that are upregulated in animal models of drug addiction and also in human drug addicts. And we came out with two of them that are called pleiotrophin and methcane that are also important survival factors for dopaminergic neurons. We have developed the methcane knockout mice and the pleiotrophin knockout mice, and we are testing the effects of the drugs on these mice in the condition-place preference paradigm, that is basically to teach the animal to associate an environmental cue to the rewarding effects of cocaine. Once the animal has acquired the cocaine-induced condition-place preference, what we do is to try to extinguish in a very demanding protocol of active extinction this cocaine-seeking behavior through 4 different extinction sessions. After that, we try to categorize the animals in the ones that extinguish cocaine-seeking behaviors and the ones that do not extinguish, and we perform proteomic studies, especially in enriched phosphoprotein samples through the IMAC technique and detection of this protein by mass spectrometry. We have developed, in this model, what we have found is that the methcane knockout mouse are the ones having more difficulties to extinguish cocaine-seeking behavior, and we have isolated a very short list of phosphoproteins that are regulated in the nucleus accumbens of this mouse, that is a brain area very related to drug reward. So we expect some of these 8 or I think 10 proteins are possibly to be evaluated as biological markers for drug relapse. That is more or less the translation of this to the human pathology. As conclusion of this talk, I would say that we have demonstrated that deficiencies in endogenous methcane facilitate the acquisition and difficult distinction of cocaine-induced place preference. Again, this is like the rewarding properties of cocaine. And also, the data suggests that methcane is a single genetic factor that is modulating the vulnerability to cocaine relapse. Our wish list, trying to acquire the imaging techniques to perform in our models, is to make more robust the use of these imaging techniques to anticipate the risk for drug relapse or to reveal increased vulnerability to the rewarding effect of drugs by finding robust correlation with biomarkers, like, for example, this methcane factor, or testing methcane PTN genetic associations to fMRI and MRI phenotypes in addicted populations. Finally, this is our building, the contact information and relevant papers from our group on this subject. Thank you very much.